We'll go ahead and get started. Welcome everybody to our first ever LTER Flash Talk Friday. Very excited to see some cool presentations in very rapid succession. This event is organized by graduate students in LTER for graduate students. So all of our presenters are students working at different sites in LTER. Today we have James, Eliot, Nicole, Emma, Ava, Rachel, Annie, and Katie presenting. So thank you guys for being here and working up some presentations for us. First, here's a quick overview of what today is going to look like. So we have four students in our first half of the presentations, and they'll each spend 5 minutes talking about a project or something at their site that's going on. And we'll let them all speak, and then we'll have a Q&A session that's 15 minutes long, where we can ask any of those four students questions about their project. And that's your chance to kind of inquire and get to know more of what you're interested in. Then after our first question session, we'll have the next four students present, and we'll have another 15 minutes. And just make sure to focus your questions to the second half of students in that second Q&A session, just so that we can kind of even out how many questions everybody gets. Oh, and then for the first half, presenters look out for an emoji reaction from Katie, who will be timing you all. She'll give you a 1 minute warning at the 4 minute mark, and then you'll know you have 1 minute left. And then for our second half, James will be our timer. So be on the lookout for an emoji reaction of some sort from him to give you that 1 minute warning. So yeah. And then also before we get started, I wanted to make sure to reiterate that we have an LTER code of conduct. So just make sure that you guys are abiding by that. Make sure to treat everyone with respect, critique ideas rather than individuals, avoid harassing behavior, and abide by the principles of academic integrity. So, do we have any questions before we get started? Okay, awesome. Then without further ado, I'll hand it off to James, who is our first speaker. You can go ahead and share your screen. And James will be presenting today from the Florida Coastal Everglades about food webs and trophic niches of the Florida Coastal Everglades. So go ahead.
All right. Thank you so much, Haley. Yeah, as Haley said, my name is James Sturges. I'm a PhD candidate in Dr. Jennifer Rehage's lab at the Florida Coastal Everglades, LTR. And today I'm going to be talking about this awesome food web project that we've kind of started and it's starting to come to fruition. So in the Everglades, we have this incredibly dynamic mosaic of marsh, mangrove, and seagrass habitats. And we know that these different habitats are connected by consumer movements, but also the hydrologic transport of biological material. And we know that these systems are subject to change because of both restoration efforts and continued press and pulse disturbances. So things like hydrologic restoration to restore the fresh water flow, but also things like sea level rise. And so, in these systems and in many coastal systems, we often think about the basal resources and the energy fueling them in terms of pathways. So, for one example, you have the green energy pathways, and these are going to be what you typically think of as fueling a food web. They're going to have shorter, more efficient food chains. They tend to have faster rates of production and turnover, meaning they assimilate into consumers faster. And then this tends to support the greater rates of secondary biomass productivity that we think of in these diverse consumer food webs. But at the same time, we also know that there's very important brown energy pathways. And so these are going to be detritively based. They tend to have more trophic transfers and less efficient steps. But we know that this is a critical way of accessing most of the fixed carbon in aquatic systems that might come from terrestrial, like organic matter and plant detritus. So in the FC, to orient you more with our site, we're really working in two major river drainages, the Shark River Slough up north, and then Taylor Slough, which is closer to the Miami Urban Center and flows directly into Florida Bay. All of this work that I do on food webs is within Everglades National Park, and that image on the right in this map kind of made by our very own Mike Ruge gives you a sense of what that habitat and ecosystem gradient kind of looks like. And then one site that we're going to talk about particular in today is RB10. This is where my lab has actually been doing long-term electrofishing at fixed sites for over 20 years. So it's ongoing work in an incredible place, and it's awesome to be a part of it. So one of the things that we've done recently is actually publish this manuscript. There's a QR code for it. Shameless plug, I know. But at these 9 food web sampling sites, we've started long-term tissue collections that go back from 2019 to the present. And now we've amassed a data set with over 2,500 rows of stable isotope data, and that's carbon, nitrogen, and sulfur. And so this represents approximately 116 different species or organic matter types across the system. And it's intentionally a very diverse collection across trophic levels and feeding gills that we can then use this isotope data to run great Bayesian mixing models to kind of estimate the consumer reliance on different energy pathways. And that's exactly what we're starting to do. So I'm just giving you a really quick, brief look at one of our 9 sites and kind of the long-term trends in energy use at the basal resource level for some of our big players. And if you were to look at that at just a pathway specific level, you can start to see that the energy channels have some trends to them, like largemouth bass and common snook being kind of our big generalist consumers, fixing energy or taking energy from both pathways. And then interesting things like fluctuations in some of our consumer populations or interesting dynamics in non-natives that also seem to be kind of more generalist in their resource use. And so one thing that I'm personally excited about is going a step further with this data and starting to build community niche hypervolumes. And so what you see here are these point clouds of those same 6 species that I just showed you at our Rookery Branch food web, and showing a community hypervolume or niche based on their resource use estimated from those trophic Bayesian or those stable isotope Bayesian mixing models. So we're starting to build this really robust way at looking at change in the trophic niche through time. And we can start to pair that with other forms of long-term data. So this figure on the left is the hypervolume size from those GIFs that I just showed you, then matching with estimates of compositional stability. This is a community trajectory analysis of those Rookery Branch food webs that we electrofish in. So the combination of both is really setting us up well to build this long-term isotope data set to understand change in a dynamic system. Our hypervolumes are going to help us develop a much better understanding of niche theory and community dynamics. And then we're starting to ask some really critical questions about the drivers of energy pathways, the community stability, and the food web structure in the Everglades. So thank you for listening.
Awesome. Thank you so much, James. That was wonderful. Okay. So next up, we have Eliot Hall, who is from the Georgia Coastal Ecosystems. And they'll be presenting sublethal predation of benthic invertebrates in southeastern estuaries. So go ahead and take it away, Eliot. You're muted still. Sorry.
So sorry. Hi, I'm Eliot Hall. I am a graduate student at UGA in the Byers Lab, and I do my research at the Georgia Coastal Ecosystem LTER, and it is on sublethal predation and benthic invertebrates. There we go. Okay. So, estuaries provide a wide variety of really important ecosystem services, and benthic invertebrates are particularly important in supporting this ecosystem functioning, not only because they're important to nutrient cycling, but also because they're very important basal resources for higher trophic levels. But generally, when we consider trophic interactions between benthic organisms and their consumers, we're generally only thinking about lethal consumption. So, what else is happening? So in benthic invertebrates, we have lethal predation, but it's also very common for them to experience sublethal predation, where a consumer takes only a piece of the benthic prey, and the prey not only survives, but is able to regrow its lost body part. So this has really interesting implications for energy dynamics in estuaries, as well as population regulation of benthic prey populations, which is the goal of my overall dissertation, to assess the role that sublethal predation is playing within estuaries. But more specifically for my first project, I wanted to determine how frequently sublethal predation is occurring Does sublethal injury facilitate lethal predation, which is seen in a lot of bivalves in particular? And how much biomass is generally being lost to sublethal predation on an individual level? So, to address these questions, I did an experiment using Mercenaria clams as a model system, which are susceptible to sublethal predation on their siphons, which you can see here, the difference between a fully intact siphon, which has a lot of dark tissue, whereas below a recently sublethally injured siphon. So I crossed these initial siphon conditions with 3 different treatments and then left them in the field for 3 days, and then upon collection, reassessed the surviving clams' siphon conditions for signs of recent sublethal injury. So this experiment was done in Fort Johnson and Charleston, South Carolina, as well as Sapelo Island, Georgia, at the GCE. So my results showed that there is roughly between an 11 to 12% chance of sublethal predation in comparison to about a 13% chance of lethal predation. So showing that lethal predation is happening roughly about the same as sublethal predation. And in a field survey that I did, I also found a really high prevalence of existing sublethal injury in clams. So this suggests that sublethal predation is happening a lot, at least to Mercenaria in southeastern estuaries. Additionally, I found that existing sublethal injury did not facilitate lethal predation. It actually decreased the chance of lethal predation, whereas it did facilitate subsequent sublethal predation. So interesting and kind of unexpected results there. And then lastly, my results showed that there was not a statistically significant difference between clams, but in biomass in clams with sublethal injury and that had not been consumed, so were still fully intact, meaning that the amount of body tissue that clams are losing to sublethal predation is very minuscule, or at least not able to be detected with the methods I used. So I want to thank you all for listening and for coming today, and special thanks to all of the support that I've gotten from the lab and the GC, LTER, and Grice Marine Lab members, my interns, and I'm very interested in considering the effects of sublethal predation in other systems as well. So if this seems like something that's interesting in your system, I would love to hear about it. Thank you.
Great job. Thanks, Eliot. Okay, next up we have Nicole Webster. So Nicole, if you can get your screen share ready. Nicole is from the northern Gulf of Alaska and will be presenting on underwater soundscapes in the Gulf of Alaska. So take it away.
Sorry, couldn't find the mute button. Are you still seeing my screen correctly?
I think so. Yes, we can see like the notes as well.
Oh, that's not right then. There we go. Okay.
Nice.
All right, so hi everybody. My name, as they've said, is Nicole Webster. I'm a PhD candidate at the University of Alaska Fairbanks with the Northern Gulf of Alaska LTER. And I'm co-advised by Seth Danielson, who's an oceanographer, and Kate Stafford, who's an acoustician. So to start off, what is a soundscape? So a soundscape is the biological, geological, and anthropogenic sounds characteristic of different geographic regions. It includes both communicative sound between organisms as well as general ambient noise that comes from non-biological sources. And variances in soundscapes are caused by differences in weather and human use, as well as community structure in the biological communities. So you're going to see a lot of these colorful graphs that I have on the right here. These are called spectrograms. They're a way to take a sound and put it into visual space so that I can visually look for patterns instead of spending hours upon hours listening to recordings. So you can see how different patterns will look very different in this system. So there are 4 sites that I'm looking at here across the Gulf of Alaska, 2 of which are continental shelf systems, so shallow-ish as far as the ocean goes, but far away from the coast. and then two bay systems, one of which is an icy fjord, so it has glaciers at the head, and one of which has been deglaciated over time. And so I'm asking here, what are the pressures and contributors to regional underwater soundscapes within the northern Gulf of Alaska? And so my dissertation as a whole looks at everything from the activity and diversity of marine mammals to diel patterns to oceanographic factors or shipping presence. But today we're just going to talk about what are the major contributors. And so the way to look at that quickly is to use these spectrographs. So you have a log scale frequency on the x-axis and power spectral density on the y-axis, which you can essentially think of as sound pressure or loudness of a sound. So when we see peaks in these lines, and then there's a line for each site here, that shows us there's something making a lot of sound at that frequency. So you can see here in the summer, we have a presence of harbor seals roaring quite a bit in our two bay sites. We have fin whales coming into our two shelf sites in the fall and winter and singing pretty constantly. We have rain as a constant signal in all of these sites in all seasons. And then in the middle frequencies, we have humpback whales and orcas contributing to the soundscape at kind of intermittent levels at all sites. Other acute signals that don't show up in an average spectra from the last site or from the last slide are things like bubble net feeding from humpback whales, which sounds different than their normal song. You also have echolocation from sperm whales, whistles occasionally from Pacific white-sided dolphins, echolocation from beluga whales. You also have air guns showing up in the system quite a bit. Those are used for oil and gas exploration. And then also these recorders are put on a mooring line with a bunch of other instruments for oceanography, and that line strums as the water moves past, so we can hear that. Chronic signals in these systems that are kind of occurring all of the time and just raising the level of noise across a broad range of frequencies in these sites are things like glacier calving in Disenchantment Bay, storms at all sites, but especially those more open water sites, and then vessel noise, so noise from cruise ships, from shipping vessels, and from small vessels that kind of come in and out of the systems. So the main takeaways here is that sound is ubiquitous but heterogeneous throughout the Gulf of Alaska, reflecting everything from marine mammals to glaciers to wind speeds to rainfall, etc. Continental shelf soundscapes are dominated by endangered baleen whales, poorly studied toothed whales, ships, and storms, whereas fjords with ice in them are dominated by harbor seal roars, ice movement, glacier calving, cruise ships, and smaller vessels. Here we do hear cetaceans or whales every once in a while, but they're pretty quiet, suggesting that they're under predation pressure. And then in our open water fjords that have less influence of glaciers, they also are dominated by harbor seal roars, but they have more big whales coming into the systems, and the rain is easier to distinguish than in other sites. That's what I've got.
Great, thank you. That was incredible. All right, our last speaker for our first half of our talks today is Emma. She works at Bonanza Creek, and Emma will be discussing abrupt permafrost thaw induces rapid shrub growth and increases soil carbon losses. So go ahead, Emma.
Thanks, Haley. Just want to take a minute to thank my awesome lab mates who helped a lot with the field work for this project. Frozen soils in the Arctic contain disproportionately large stores of soil carbon relative to their land surface area. And because the Arctic's warming really fast, this carbon is susceptible to decomposition and emission to the atmosphere as greenhouse gases, which can accelerate climate change. And the permafrost region itself is not responding uniformly to warming. So some areas there are massive stores of ground ice. And when that ice melts, the ground collapses in a catastrophic way, like this thermo-erosion gully that I've pictured on this slide. And these features are termed abrupt thaw features, and they're only likely to occur in 1/5 of the permafrost region, but they could create up to half of permafrost carbon emissions. And these features are also really challenging to measure, especially with traditional carbon monitoring methods like eddy flux towers. And so to better understand these potential hotspots of abrupt thaw, of carbon release from abrupt thaw, we measured ecosystem carbon cycling changes at 5 features in the west side of Denali National Park. And first, we did a 10-year resurvey of transects that crossed through these abrupt thaw features. And so these transects start in relatively flat control tundra, and they move into these collapsed gullying parts of the landscape. And we surveyed these in 2013 and again in 2023. In these plots, I'm showing shrub height in 2013 in blue and then in red in 2023. And you can see that the control tundra shrub height increased by about 8 centimeters during these 10 years. But within these abrupt thaw features, shrub height increased 5 times faster. So we had 40 centimeters of growth. And shrub height here is really important because this woody vegetation stores more carbon as biomass than the rest of the tundra. And so we can think of shrub growth as really increasing above ground carbon storage. And so these really the rapid growth that's happening within these abrupt thaw features indicates more carbon uptake by plants. We also did an intensive soil survey at one of these sites where we directly measured soil carbon stocks. This was at the Wigan Creek site. And this site is a thermo-erosion gully, and I'm going to show you how the gully evolved over time. So for reference, keep an eye on this pond here. This imagery is from 2004. And in 12 years, you'll notice that the gully expanded and it's more spiderwebby and it has more lines that resulted from ice wedge melt. And by 2024, this pond has just drained and become part of the rest of the thaw terrain. And so we sampled soils across this disturbance using a space for time approach. And what that means is that sites in the control tundra represent undisturbed areas. And then as you move closer to this stream, the sites here are closer to where the thaw initiated. And so they're experiencing disturbance for longer. And at these sites, we measured soil carbon stocks using soil cores. And this was to understand how abrupt thaw changed below ground carbon storage. And what we found is that across the whole soil profile, this is to about 55 centimeters, there was a significant loss of carbon between the control and the site that had been disturbed the longest ago. So it's next to the gully, and then also directly within the gully where all of the ice had melted. So this equates to about a 40% loss of carbon on the side of the gully and in the most disturbed area, and a 60% loss of carbon within the gully. And so really we're seeing a trend of more disturbance increasing carbon losses. And so these results show both a rapid growth of shrubs and also significant soil carbon losses, which means that abrupt thaw is increasing both above ground carbon uptake and below ground carbon losses. But really importantly, the total carbon stored in biomass above ground is orders of magnitude lower than the carbon losses that we measured. So this biomass estimate is about, for this kind of shrub tundra, is about 0.4 kilograms of carbon per meter squared versus the 10 to 15 kilograms of carbon per meter squared that we lost. And this is that main takeaway summarized. So we found that it's really highly likely that abrupt thaw features are going to be significant carbon sources because plant carbon is unlikely to be completely offset by soil carbon losses from permafrost thaw. And that's all I have.
Wonderful, thank you. Okay, that is it for our first half of our talks. Thank you, everyone. Those were all amazing talks. And now I'll open the floor to anyone that wants to ask questions from James, Eliot, Nicole, or Emma about their presentations. Yeah, Namir.
Sure, yeah. Thank you all for these talks. It was really cool to see what y'all have been up to. I mean, I guess I had questions for everyone. I'll just ask one right now. But going back to James's talk, I was wondering, you had some interesting figures at the end there showing that there was change in the fish community over time. I wonder if you could speak to like what are those potential drivers in the change in the fish community that you're seeing, and then how you're hoping to be able to track that through the isotopic data and those samplings that you got.
Yeah, so great question. Our long-term time series comes from just the core fish in our Rookery branch site, and that's been changing upstream because of freshwater kind of coming back into the system in Shark River, kind of removing barricades to flow and improving that. At the same time, we're also seeing change more recently, and it might pair well with the food web data due to invasive species. So we've seen boom bust cycles of peacock eels that have definitely caused some instability in the system. And now more recently, there's concern in the marsh of Asian swamp eels moving in. And for the first time in the 23-year of the data set, we detected them in 2023, and then this year had more than the previous years combined. So community-wise, we're seeing change. Um, Condition-wise, we're also seeing change, and it'd be great to start, like you said, pairing isotope data and kind of putting those pieces of the puzzle together to understand the change in the system. Thank you for your question, Amir.
Awesome, thanks.
Yeah, Marty.
Sure. Great talks, everyone. Really appreciate it. For Emma, um, clearly there's a lot going on within those abrupt thaw areas. Do you have an estimate of how much area that is right now and how that is likely to increase or decrease over time?
That's a good question. The feature that I showed is about 1 kilometer squared, so it's pretty small. And a lot of work, I think, for the detection of thaw features focuses on retrogressive thaw slumps because there are these huge, massive ground collapse features that you can see from satellite imagery. It's pretty hard to detect something that's this small, like a gully that's this small. But I think that like that's probably the dominant response from the land. So there's definitely a huge gap there. But I know that the other challenge is that a lot of these features are formed where there's high ground ice content. And so that's something that people are focusing on mapping. But it's also challenging because it's below ground. So it's definitely a big unknown, and that's kind of why it's a big unknown in terms of the carbon budget too. So we don't really know how much like the estimate of 50% of carbon emissions comes from a model. So yeah, it's going to increase because temperatures are increasing, but I don't know how much.
Thanks.
Someone should do a PhD on that.
Good.
I actually had a question for Nicole. It's kind of more of a methodology question, but do you ever get two sounds that have the same frequency, and how do you kind of decipher between the two when you're, trying to figure out which sounds are which in your graphs?
Yeah, that's a good question. So I would say if it's something like a marine mammal, they're pretty distinct. It's really easy to tell a killer whale from a fin whale, for example. They're completely different frequency ranges. But something like dolphins in other parts of the world are still not distinguishable by sound down to species. So you just say, well, it's in this group, and we hope for the best. Um, my area luckily doesn't have that overlap, but things like at the low frequencies that are more chronic signals, so things like ships and ice noise, are harder to distinguish. So I can't say definitively all the time, for example, what is ice and what is a harbor seal roar, because if they're both far away, they both just sound like rumbles. And so that is a case where I pull out those those spectral lines because they can tell me the seasonality of a feature. So whereas I expect a glacier to calve at least somewhat all year long, I only expect harbor seals to roar in the summertime when they're breeding. So you have to pull in other context clues basically to make an educated guess as to those things.
Cool, thanks. Any other questions for our speakers? Feel free to throw things in the chat as well. I see something from Marty. Oh, go ahead.
Yes. Yeah, Marty. So that is next steps in my data. The first steps was to establish who is producing sound. And the next steps are to start training things like neural networks to actually comb through my years of data and say where XYZ whales are, because doing it by hand is incredibly time consuming. But developing different models for each of those whales is also time consuming. So it's a process.
Yeah, Namir, go ahead.
Yeah, I had a question for Elliot as well, and I was wondering, like, it's really cool data seeing that sublethal predation leads to more sublethal predation, but not to more actual lethal predation. And it, like, obviously that's can be adapt seeing how that's adaptive from the thing that's being consumed side. But I'm wondering, like, is there a hypothesized mechanism for how that works from the predation side? Like, are these injured nubs, do they look more tasty than like, or like what, how is it that the predator is potentially being taken advantage of here, or at least being directed to something that's not as damaging to the clams? That part of the interaction was really interesting to me.
Yeah, good question. So identifying sublethal predators is also something that's a little shaky, especially in estuaries where um, we don't have the luxury of always having the most effective like underwater videos, for example. But it's been suggested that sublethal predation is particularly important for smaller size classes of consumers and juveniles of pelagic species, which again, in the context of estuaries functioning as nurseries for a lot of marine species is really interesting. There's also kind of a I don't want to say a contingency, but in sublethal predation, you can consider it a foraging strategy, which is how I'm considering it a lot. But there are also cases of sublethal injury happening as a result of a failed lethal predation attempt, which would leave a lot different visual evidence, I would argue. But that is another case of sublethal predation where you could take this view of, well, the predator or the consumer is getting snubbed of energetic value, so to speak, versus if it's a smaller consumer and it's a foraging strategy, then it is getting kind of a equivalent energy input.
Yeah, that's very cool. I mean, thinking about the things being consumed as kind of like plants in a way is just,
Mhm.
yeah, is a very different take on how to think of predation. So interesting. Thank
Yes,
you.
a lot of overlap with like herbivory and parasitism, things like that.
Okay, do we have any other questions for our speakers? Feel free to raise your hand or type something out in the chat. Okay, if not, then I guess we can move on to our second group. Thank you guys for presenting our first half. So our first presenter in our second half presentations is Ava. They'll be presenting from Cedar Creek about differences in carbon accrual between forest and grassland soils. So go ahead and take it away.
Okay, hi everyone. Sorry, I had trouble. I also had trouble finding the unmute button there for a second. Okay, hey, it's really great to be here with you all today. My name is Eva Schwarz. I'm a second year PhD student at the University of Minnesota, and I am doing some work at Cedar Creek LTER on how soil carbon accrues differently between different habitat types at depth. Um, so one really cool piece of research that has come out of Cedar Creek LTER is that um, more diverse plant communities are more productive, so they make more biomass. And more plant biomass means more dead plant litter on the ground at the end of the season. And since plants are made of carbon as well as a bunch of other things, that means more opportunity for carbon to be stored in the soil. Um, and we know that the majority of uh, soil carbon in the world is stored below just that top 20 centimeters that so often gets sampled. We also know that soil carbon that is stored deeper in the soil is stored longer and more stably. And so it's important to understand whether and how plant biodiversity impacts soil carbon storage at depth. And it's also important to know whether that differs between ecosystem types. So, for instance, between grasslands and forests. And so, lucky for us at Cedar Creek, we have not one, not two, but four different plant biodiversity experiments. So they're all shown on this map here. Over here on the left are two grassland biodiversity experiments, Biocon and Big Bio. And these are both places where you can see these different experimental community squares here in Big Bio. Each one of those is a different plant community with a different number of species in it. And the same is true of our FAB, so forest and biodiversity experiments. There's FAB1 is much smaller, it's got smaller plots, and then FAB2 is these kind of bigger forests. And each of these squares, again, is a different tree community with a different number of tree species in it. This photo was taken when FAB2 was very young, so I promise you these look much more like adult trees now. And thanks to long-term LTER data, we have decades of soil carbon data on Biocon, Big Bio, and FAB1. And this past summer, we just took the 10-year data set of soil carbon data from FAB2. So we're now able to compare soil carbon dynamics across the first decade of each of these 4 plant biodiversity experiments. And so what we found, to orient you to this graph, on the y-axis, we have yearly gain in percent soil carbon. So per year in that first decade, how much did each of these soils gain in percent soil carbon? And then along the x-axis, we have depth. So the depth intervals are a little bit different between each of these experiments, but generally they go down to 60 centimeters in 3 more or less even bins. And we have the graphs faceted by the 4 different experiments. So over here on the left are our 2 grassland experiments, Big Bio and Biocon, and over on the right are our 2 forest biodiversity experiments, FAB1 and FAB2. And then the diversity treatments are shown in color. So the pink boxes are the monocultures, which have just 1 species. And then the blue boxes are the polycultures that have multiple species. So it's 12 or 16, again, depending on which experiment we're talking about. And so what we found was that in the first decade, diversity increased percent soil carbon in grasslands, but only in the top 20 centimeters. So again, you can see that these blue boxes representing the diverse plots had a higher yearly gain in percent soil carbon than the monocultures. But this was only true in the first 20 centimeters of those plots. So you don't really see the same effect at those deeper depths yet. And then we had some interesting contrasting results in the forest experiments. So percent soil carbon also increased in diverse plots in the top depth of one of our forest experiments, but not in the second forest experiment. And you might be wondering, why would 2 very similar experiments right next to each other have very different results in terms of what's happening with their soil carbon? And I'm going to give you the short answer, which is that we think it's grass. This is my fellow grad student, Alex Griffin, measuring the understory community of FAB2. And I'll just say for now that the big difference between these experiments is that FAB1 really doesn't have much of an understory because it's canopy closed very early on, whereas FAB2 has a fairly diverse understory. And so we think that those understory plants may be swamping out any diversity effects that we see from the tree diversity, because, you know, even if you have a plot that's a monoculture of birch, it's not really a monoculture. There's actually tons of different species in there. And yeah, I will leave it at that. Thank you all for being here today and listening, and a huge thanks to the data collection crew and to the Cedar Creek staff for making this possible. Thanks, everyone.
Thanks, Eva. That was great. All right. Our next presenter is Rachel from the Kellogg Biological Station, and Rachel will be presenting on soil health gains under KBS LTER prairie strips. So Rachel, whenever you're ready, you can go ahead and get started.
Sounds good. Thank you. Hello, everybody. I'm really excited to be here today. My name is Rachel Drobnak. I'm a current PhD candidate at Michigan State. My lab with Christine Sprunger is based at Michigan State University's Kellogg Biological Station. And like my title suggests, I'm really excited to share with you all how established prairie strips in the LTER build soil health. So if you're unaware, Eva set us up really nicely. Prairie strips are an agricultural conservation practice that focuses on increasing above ground plant diversity in agricultural landscapes. And so farmers are encouraged to convert portions of monoculture row crop fields into perennial native grass restoration areas. And work done at the KBS LTER so far shows that prairie strips do a lot to provide multiple ecosystem services, such as increasing insect abundance and pollination above ground, really diverging and creating unique below ground bacterial and fungal communities. And one of the most open-ended questions so far is what is going on below ground in the soil? And early on in this experiment, we found that after the first 3 years of establishing prairie strips, the soils really only had variable increases in soil health represented through quantifying soil carbon, and that this really depended on crop type and carbon indicator. So this leaves an open-ended question like, so what is going on now with prairie strips below ground as they continue to age after 3 years? And that's a question that I get to ask as part of my PhD, which is, can prairie strips build soil health under previously cropped land, and how long does this take? And so to answer this question, I am unable to measure really large general pockets of soil health, such as quantifying total soil organic matter, because it's been shown that soil organic matter takes decades to change. Instead, I focus on components of soil organic matter that can be quantified through specific soil health indicators, I call them. These indicators can quantify fractions of soil organic matter, which have been shown to change more rapidly over time and can give us good insight into what's happening below ground without needing to wait decades and decades to detect overall trends. So the indicators I'm going to show results for today are soil protein, shown in purple. This measures a fraction of soil organic nitrogen in the soil. Permanganate oxidizable carbon, or POC, which is an early indicator of carbon sequestration or carbon storage. And then mineralizable carbon quantifies both how much microbial respiration is occurring in the soil and potentially how much food source they have from labile carbon. So this work took place at the main cropping systems experiment at KBS. I sampled 2 of the annual row crop treatments, which have received prairie strips since 2019. These treatments are shown in green and blue. The chemical input system shown in green is an annual row crop rotation, which has received an input of inorganic fertilizer and pesticides. The biologically based system has never received an input of fertilizer or pesticides. And we took samples, over 700 samples over the course of 4 years in the prairie strips experiment, from 2022, when the prairie strips were 3 years old, until 2025, when they turned 6 years old. If you were to go out and look at them, they'd look kind of like this. We took samples in the prairie strip, shown in the green dot, and then at 3 distances into the adjacent cropland. And for today's results, I'll be aggregating all of those cropland distances together because we saw inconsistent significant differences between them. All right, so do prairie strips build soil health, and how long does this take? So I'm first going to show results from our fertilized treatment. That's the chemical input cropping system for soil protein. Results increase starting in the 4th year after establishment, which is really exciting. And this difference between prairie and cropland soil protein continues to increase over time. In our unfertilized treatment, differences between prairie and cropland soil protein takes until the 6th year after establishment to detect consistent changes. With our other indicators, POC had an identical trend where in a fertilized cropping system, protein or POC increased after the 4th year, and then in our biologically based system, this happened in the 6th year. And finally, with our most sensitive indicator, mineralizable carbon, we started to see increases in the prairie strips starting in the 3rd year in the no input cropping system in our biologically based system. And these differences increase over time and even become more drastic within each year as the prairie strips get older. All right, so I'll just end by saying we can kind of say now definitively that prairie strips do increase multiple measures of soil health after 4 years in systems that receive fertilizer and 6 years in systems that are unfertilized. Different indicators take somewhat different amounts of time to detect changes. All right, thank you so much.
Awesome. Super interesting. Thank you so much, Rachel. Okay, Our next presenter is Annie from Niwot Ridge. And Annie will be talking about friends for dry times as growing seasons lengthen, mycorrhizae emerge as late season mutualists in alpine soils. So Annie, go ahead and
Oh
get
gosh,
started.
spoiler
Ah,
alert, you guys. Sorry. Um, Sorry, I have a frog in my throat. Well, hi, my name is Annie Meeder. I'm a PhD student at University of Colorado Boulder. I work at Niwot. I'm originally from California. I grew up about 12 minutes from the beach, and I've only seen snow once. So when I came to Colorado to do research in the Alpine, I was a little bit scared. Um, but I had a really great team, and we were really determined to understand how life survives in one of the harshest environments on Earth under climate change pressures. And one of the things that I wanted to study is alpine plants. And alpine plants don't survive alone. Beneath the surface, they're connected to a hidden world of fungi, which are partners that help them access nutrients, retain water, and keep growing under extreme conditions. And so the question that I wanted to ask was, when climate change shifts plant timing, do the root fungi keep up? So, in these nutrient-poor alpine soils, plants rely on root-associated fungi, and there are two species on the left that we typically see in the roots. These fungi trade nutrients like phosphorus and nitrogen for carbon from the plant and can also improve water uptake. And these partnerships can be essential for plants in harsh environments. But the alpine environment is changing rapidly, and early snowmelt is shifting when water and nutrients are available. So, my question is, are these plant fungal partnerships flexible enough to keep up with climate change? And so, specifically, we expect warming from climate change to cause early snowmelt in the alpine. To test this, we simulated climate change directly in the alpine at Niwot Ridge LTER. We added black sand to the snow surface, which absorbs sunlight and causes the snow to melt about 7 to 10 days earlier. And so my first step was to make sure that this experiment was successful. Um, and so the black sand treatment did successfully change snowmelt timing. So we had an early snowmelt treatment. And that was also influenced by snow depth and topography. And sorry for the weird spacing. PowerPoint was struggling this morning. And so the next step to this was to test how plant fungal relationships responded to our simulated early snowmelt experiment. So we collected phenology information from 5 different plant species, as well as their plant roots for fungal analyses. And the first thing that we needed to do was see if their plant phenology changed. And so earlier snowmelt does strongly change plant timing, especially when flowering begins, and when plants set seed. And so the shift timing and timing carried forward into reproduction and so root fungi can strongly respond to phenology shifts in plants. And so that was something that I wanted to look at because I did find that fungi was not responding to changes in snowmelt timing. But they did actually respond to these shifts in when plants were flowering and setting seed. And they would increase if a plant was setting seed or flowering later. And so I found that really interesting because now I can ask, where do fungi fit into this system? And what is actually driving this specific response? And so one of the things that I tested is early snowmelt shifts phosphorus availability across the season. And so what this is showing is that in plots where snow melts earlier, phosphorus is more limited across the season, because as soil is dry, phosphorus gets locked up in the soil. And so by late in the season in early snowmelt areas, when plants need phosphorus the most for seed production, there's not a lot available. And so this creates a sort of nutrient bottleneck. And I think that the plant's response to this nutrient bottleneck is actually to recruit fungi in their roots. And so, to give you some takeaways, climate change is shifting snowmelt timing, which reduces phosphorus availability, and in response, alpine plants are likely relying more on fungal partnerships. And these partnerships seem resilient right now, but they may be pushed to their limits if the alpine continues to dry. And with that, thank you.
Great job. Thanks, Annie. Okay, and last but certainly not least, we have our final presenter, Katie, also from Niwot, who will be presenting on exploring plant responses to changes in climate using a large-scale turf transplant experiment. So, Katie, take it away.
Thanks, Haley. So like Haley said, my name is Katie Bardsley, and I'm a PhD student at the University of Colorado Boulder. And like Annie, I'm part of the Niwot Ridge LTER. And today I'll be telling you about my research using a turf transplant experiment to investigate demographic responses to warming for two species of alpine forbs. So Annie already introduced us to this site a little bit, but to situate us geographically, Niwot is located in the southern Rocky Mountains in Colorado and is a high elevation site that spans from the Continental Divide to subalpine forest. And in the fall of 2024, the Niwot team set up a turf transplant experiment at this site, which is the main focus of the research I'll be telling you about today. And in contrast to some of the other projects we've heard about, this project is still fairly early stage, so I'll be focusing more on what we're doing rather than results. So this experiment was designed to enable us to investigate responses to warming across scales at population, community, and ecosystem levels by transplanting intact plots of plant and soil or turfs between sites that have a 3 to 4 degrees Celsius difference in growing season soil temperature. And this design was replicated in dry and moist meadow communities. And the images and time lapse here give you a sense of what these transplant efforts looked like. We transplanted 384 turfs, and it was a lot of work for a large team of people. To provide a little more detail about our experimental design, we have 3 different transplant treatments within a given plot. So we have local controls, which have never left the ground, local transplants that were dug up and moved within a given site. And then we have non-local transplants that we dug up and then moved between sites that have a different temperature, but within the same community type. And then within these treatments, we have 1 replicate each for intact monitoring, destructive soil sampling, and plant thinning, and the last of which is designed to investigate how species interactions impact responses to warming. Given this design, we were able to study the impacts of a novel temperature regime from a variety of angles, including the ones I have listed here. So there are a lot of different teams and people involved in these efforts. So we have entomologists, plant ecologists, biogeochemists, and it's a fun group to work with. So my main focus is the plant population demography part of things. And I'm focusing on 2 species of bistorts, Bistorta vivipara and Bistorta bistortoides. And one key difference between these closely related species is their reproductive strategy. So Bistorta vivipara on the left primarily reproduces asexually via bulbils, while Bistorta bistortoides exclusively reproduces sexually. And while their ranges overlap in the mountainous regions of the western US, they differ slightly in the climatic spaces they occupy. So Bistorta vivipara is more common in drier, cooler sites than is bistortoides. So not only is this true for the ranges across North America, but the trend also holds in plots I've sampled at this site. So here I'm showing preliminary data for the mean number of individuals per subplot by species and site. So we see that Bistorta vivipara in blue is much more common in the dry meadow, especially at cool sites, where the opposite is true for Bistorta bistortoides in red, which is more prevalent in the moist meadow and more prevalent at the warmer site than the cool site. So I'm tracking every individual of both species within the experiment, represented by the preliminary data here, to address 2 main research questions. How does an altered temperature regime influence population dynamics? And what are the roles of phenotypic plasticity and genetic variation in shaping demographic responses? And I plan to collect 2 more field seasons worth of demographic data with a focus on tracking individuals and measuring their size and reproductive output for a total of 3 post-transplant field seasons. And then I'll use that data to investigate demographic patterns as a function of species and their environment. So with that, I'd like to thank the whole Niwot turf transplant team. As I mentioned, it's a lot of work to get this experiment set up, and there's a lot of people involved. So big thanks to everyone, especially those who have helped with data collection and the transplant efforts.
Great. Thanks so much, Katie. Okay, and that was our last presentation for today. So I will open the floor to anyone that has questions for Eva, Rachel, Annie, or Katie. Feel free to unmute yourself and ask or type things out in the chat. I can start us off, actually. I have a question for Katie. How far apart are your like local and non-local transplant sites from each other? And like how big are the sites themselves? So like what's the magnitude of difference, I guess, between like local versus non-local?
Yeah, so I think it's about a little over a mile between the sites that are furthest apart, roughly. And the part of the difference in the temperature comes from differences in elevation and aspect. So like how the sun is hitting the sites. And then like when you're standing at one site, you can like see all the subplots within that site, or the plots within that site. So they're pretty close together within a site, and then it's like it can be up to that like mile, a little more between the non-local.
Gotcha. Thanks. Emma, you have a question?
Yeah, I have a question for Rachel. I was curious, you mentioned that you grouped all of the cropping system data together. But did you see any, maybe they weren't statistically significant, but did you see any effects of the prairie strips on like immediate crop soils? Like I missed your slide, or yeah, anyway, maybe you addressed this,
Yeah.
but
Yeah, no, great question. So we sampled 1 meter from the edge of the prairie strip, which would be the closest distance, and then 5 and 20 meters. I would say in terms of the three variables I presented today, there were no consistent effects over any of the sampling time points, even at 1 meter away, for these three soil health variables. I think the mechanism of these variables changing really has to do with like direct above ground plant and then below ground rhizosphere communities. So not I don't know how far like horizontal roots from the prairies are extending into the prairie strips, but at least my data are showing that that doesn't have a large effect on soil organic matter processing. For other indicators, though, I think that there is potential for spillover below ground. For example, uh, our lab studies below ground free living nematodes, and previous work in other strip systems, we've found that there is spillover below ground. So, yeah, that's kind of a next step for me.
cool, thanks.
Yeah, Namir.
All right, awesome. Thank you all for all those presentations. Really, really cool work. I had a question for Annie, and it might not be relevant given the system because I don't work in alpine systems, but I know there's some evidence out there that fungi are also can help plants obtain water, or they could be potentially their hyphae could be translocating water along the outside, and that would also potentially match up with the timing of when your phosphorus demand would tie in there. So my question was just like, if that something that happens in alpine soils, or is that something y'all are considering at all? And yeah, that's basically it.
Yeah, I think that's a really good question. Definitely considering that. I wasn't able to necessarily, the only issue with this experiment is the thing that I was able to directly say is fungi are responding to plant phenology, not snowmelt, and sort of suggest, like, okay, here are some trends that we're seeing. Maybe this is why. And so, like, that is another reason why, like, the soils are drying out. Also, there's like nutrient limitations, and that tends to be when plants recruit more fungi. So I can't exactly give you an answer to that, but I can tell you that that is evidence that I am thinking about. So Thank you.
Awesome. Thank you.
Can I ask a question to Eva? I had a question for you. I was curious with the long-term like forest biodiversity experiments. It maybe just because it hasn't been going on for as long, but I was surprised that they had less of a drastic difference in whether it was a polyculture or monoculture versus the like yeah, like the grassland experiment. Do you have any hypotheses about what like the magnitude of the trends you saw comparing both habitat types?
Yeah, totally. Great question. Thanks. Yeah, so it's something we're thinking about a lot. One of the biggest things is that the forest plots were planted into an area that was grassland beforehand, whereas the grassland was also planted into something that was grassland beforehand. And so um, with the forest biodiversity experiments, we've been starting to think about it more in an afforestation context as opposed to like what's up in like regular forest systems. It's more like what happens to soil carbon dynamics when we try to start planting trees in something that didn't used to be trees. Um, and so, one of our sort of hypotheses around that is that as you start planting trees into grasslands, instead of um, necessarily having any strong diversity effects of the trees. You kind of just are slowly replacing grass biomass with tree biomass. And again, in the even in the forest plots where we did see an effect of biodiversity, there are still grasses in the understory, which might be kind of swapping out some of those tree diversity impacts. And so yeah, I don't have a like 100% this is what's happening question, but those are my best guesses as to what's going on there. Yeah, thanks a lot for the question.
Awesome, thanks.
Great. Do we have any other questions for our speakers today? Okay, wonderful. Well, with that, if we don't have any other questions, then I think that's all we have for today. I'd like to thank everyone just for coming. This was so awesome. We had a really good turnout. We had great presenters. Thank you to everyone that presented today. And thanks a lot, especially to Marty and the LTER grad reps, especially Katie and James, who helped organize this. So yeah, thanks to everybody.
Thanks for hosting, Haley.